just going to do a quick fire video on simple genetics, see if we can help some beginners understand how these colour morphs are born, if they've got a pair that are different, what they're going to end up with, all the rest of it, you know. So we'll do a little case study. Let's pretend I'm back in 1975 walking around Florida, around a place called Lake Okeechobee, and I look underneath the rock and lo and behold there's an albino corn snake. Well, it's the first one ever found. Fabulous. I'm super excited. Now this animal's absolutely knackered from a natural perspective because it has no camouflage. The predators are going to come and eat it in very quick order. So I keep this snake. I've got a collection of normal corn snakes at home. So what I'm going to do is I'm going to raise it, and then I'm going to see the way that the genetics work. Now, when we start talking about genetics, there are a couple of words that are going to come up whilst you're doing your reading and your research. And, you know, scientists use these big long words and people get put off by it, but actually simple recessive genetics isn't particularly complicated. Generally, they've got two genes, which we call alleles, and they're going to be represented by a letter. So these letters, depending on whether they're uppercase or lowercase, represent whether they are a dominant or recessive gene. In the case of two capital A's, this represents a homozygous animal, which means the same, homozygous, the same. And then heterozygous means different. It means that I've got an uppercase and a lowercase letter. Because the albino, or the colour phase, is represented by two of the same size letters, that's also called homozygous. The meanings of these are homozygous 2 double A's is wild type. Heterozygous or carrier is A. Homozygous for the colour mutation or pattern mutation is represented as A. To be able to work out how these corn snakes in our case study are going to be able to affect one another colour-wise, I need to use a tool. And the tool that I'm going to use is called a Punnett square. Dead simple, 4 boxes. We add a little more information. We've got the male's genes, we've got the female's genes. The male from my private collection is AA. The female I collected 3 years earlier is now fully grown and ready to breed, and she is represented as AA. What we then need to be able to do is use the genes to work out the probabilities. Each of these squares represents a 25% probability of this occurrence. So we take the male's gene, A, and we drag the female's gene, A, into this box. And that 25% is a het. So that means a carrier. Visually it's a normal snake, but it is secretly carrying the gene for albino. As we start working our way through and dragging our pairs of letters, what becomes apparent is that by breeding my normal corn snake to the albino corn snake that I caught, every baby in each 25% block is a het or carrier. What I've then got to do, because I'm now frustrated, I've bred these snakes together, nothing's happened. So I've now got to take 2 of these babies, a boy and a girl, raise them for another 3 years, and I'm going to breed those together. So what we do with the boy and the girl of the babies we produced, we put their genes in the Punnett square edges, and we've got A, because both of the animals that were born were carriers. By breeding these together, we then pair up the genes. So we've got A and A comes together to make 25% normal snakes. And we continue doing this. The square we're most interested in is finally, after 6 years of breeding trials, I have now got 25% of the litter that will be visual albino. Fabulous. I've managed to reproduce the original gene from 6 years earlier. There's a problem though now, and this is where we see it in shops a lot, and you'll see it written on boxes when you go to the shows. What we do is, because they're albino, we take them away. We remove them. They're visually different. We know that they're albinos. What we're left with on the other 3 squares are all animals that look like normal snakes. And what we say is, of those, 66% are possibly het for the colour. 25% are normal, 25% het, 25% het. We've removed this, so what's left becomes 100%. Of that 100%, 2/3 are het, or 66%. This is the way that we're going to reproduce our albino. Very, very simple. There's a lot of writing online that you can find that will take you through the basics. Things become infinitely more complicated when we've got two genes. So say, for example, we have a snow cone, which is albino and anarthristic. Right. That suddenly then means that we need a Punnett square with 16 squares. And the snow cone is a 1 in 16 chance animal. This only works, this example, with recessive genes. There are animals out there, particularly royal pythons, which have got something called codominant genetics, which work in a slightly different way. We still have wild type, which is represented as AA. And then we have the heterozygous, but now rather than just being a carrier and it being a secret, the heterozygous animal is visually different. What that means then is there must be another colour type, which is the full AA. So, an example of this. Wild type royal python, lesser platinum royal python, blue-eyed leucistic royal python. So that's the way that the codominance carries on. The AA, or full gene, where we've got both alleles the same, that is your blue-eyed leucistic. And if the blue-eyed leucistic is bred back to our wild type royal python, we know from the previous sheets that every animal subsequently will be born a lesser platinum. This is the way that codominant genetics works. Research is important, especially when you're looking at colours and you want to know what you're going to be able to produce. Genetics aren't overly complicated, and as long as we can get our heads around these alleles, what they represent and the way that they're written, all it's a case of doing is filling in the blanks in the box. Maybe take the time, if you've got an albino corn and an anarthristic corn, to sit down. You can look online how to draw a 16 box Punnett square, and we can then extrapolate from that what everybody is who's born. And there will be probabilities in there, but as long as we can understand what the letters mean, it's easy for us to infer what we're going to end up with. I hope this has been useful. We keep trying to do little bits just to help you with the education side of things. We'll keep producing the videos. Visit this website, which is snakesnadders.co.uk, to see what we're all about. Cheers.